So we live in a microbial world. If we look at the numbers of organisms of different types on our planet, we get some interesting numbers. We know that the population of humans is about 8, 9 billion at the moment, which comes to about 10 to the power of 10. And this is sounds like a large number, but it really is not. The number of insects on our planet is estimated to be somewhere in the 10 power 80, which is about 100 million times the number of humans. And if we go down the size scale and look at bacteria, microscopic organisms visible only through a microscope, then we estimate that the number of these organisms on our planet would be somewhere in the 10 power 29 to 10 power 30, which is 1 followed by 29, 30 zeros, whatever that number is called. So uh, the the world is basically full of these organisms. And they're found in pretty much any conceivable habitat on the planet, even in the polar ice caps, in hot hydrothermal vents. And though these are extreme habitats, the sort of environments where these are found in very large numbers are usually subterranean and subsurface in the ocean. So these are obviously very, very large numbers. And this could mean one of two things. One is that these are very, very, very large number of um, organism of the same type, or they also represent a very large diversity in terms of traits as well as in terms of their genetic makeup. If we look at a tree of life, which is also called a phylogeny, um, which today typically represents genetic diversity of organisms on the planet, you'll see that there are bacteria, there are eukaryotes, that includes us, insects, fungi, and there are archaea, which are well, um, also similar to bacteria in some ways and also similar to eukaryotes in some other ways, but they are prokaryotes in that in terms of cell structure, they're a bit more similar to bacteria. And the phylogenetic space that we have is dominated by bacteria. And these organisms today, we know, are responsible for um, sustaining life on our planet. They drive biogeochemical cycles, and it's now become a cliche to say that there are more bacterial cells on the human body than human cells, and every day we discover their role in some process or the other that we thought has been uniquely human or uniquely animal, but might in fact be driven by the bacterial inhabitants of these organisms. In the 1960s, Linus Pauling and Zuckerland, they proposed that, okay, now we've come a long way and we are able to sequence or read the sequence of amino acids or bases that form proteins or DNA. In other words, we have ways to read the genetic material and their products. And evolution really operates at the level of these molecules. So can we use the sequences that of a DNA or proteins that we are now able to read to build phylogeny? So that was the idea that they had proposed. And Carl Woese, who had been working on the evolution of the protein synthesis system, uh, took up this challenge and said, "Hey, this idea of prokaryotes versus eukaryotes is not a phylogenetic classification, but instead we'll have to think about a tripartite tree of life. You have bacteria, you have eukaryotes, and you also have this other thing called archaea, Archaea are also prokaryotes, but they seem to be pretty much as different from their sister prokaryotes, that is bacteria, as they are from eukaryotes. So prokaryote, eukaryote, yes, it is a kind of classification, but it is not phylogenetic. Phylogenetically, we're dealing with a tripartite um, um, tripartite world. Of course, things have come a long way now. There are schools of thought that believe that eukaryotes are in fact derived from archaea, and especially this interestingly named Asgard archaea after Norse mythology. And we also recognize that eukaryotes are probably a product of a fusion of archaeal and bacterial cells. So eukaryotic genes would share ancestry with archaea and bacteria. But where we stand at this point, this is towards the end of the 1970s or so, is that the prokaryotes are somewhat different from eukaryotes in terms of their cellular properties, but phylogenetically prokaryotes are not a single clade of life, but instead there are two clades of prokaryotes, one bacteria and archaea, and eukaryotes are a separate clade, and the archaeal clade of prokaryotes might be as different from from bacteria as they are from eukaryotes. Okay, so fast forward a few decades to today, and if you look at any database of biological genomic information, you'll find that have several million genomes of bacteria, and these correspond to then 100,000 species. Um, and of all hues and colors, pathogenic bacteria, non-pathogenic bacteria, bacteria that can do well in very limited sets of conditions, to bacteria that can pretty much live um, anywhere under the sun and under the moon or wherever. Um, and uh, and all of this has accumulated over a period of about 30 years since the first bacterial genome was sequenced in the mid 1990s. And the huge explosion happened with the developments in technologies that happened about 15 years ago, say around 2000, late 2000s. Um, so, with all these genomes, what have we learned? We've learned that bacteria are extraordinarily genetically diverse. If I were to just look at something as simple as genome size, I'll find that bacterial genomes can be as small as 100,000 bases to as large as 12 or 13 million bases. That represents about two orders of magnitude difference in genome sizes. You can argue that the genome sizes of eukaryotes spans a much larger um, width. For example, genomes of yeasts can be maybe 10 megabases, and genomes of things like certain fishes and so on can run to hundreds of billions of bases. A much larger breadth of genome sizes than you see in bacteria. But where bacteria distinguish themselves from the eukaryotes, and especially higher eukaryotes, is in being able to pack their genetic material with a lot of information or genes. So bacterial genomes are essentially gene with little bit space for useful um, uh, uh, um, um, regulatory regions that help decide which gene is expressed to produce a protein or some other functional RNA when. However, in higher eukaryotes, they do, for various reasons, accumulate a lot of so-called junk DNA, which are largely non-functional. So even though a eukaryotic genome may span a few orders of magnitude, the numbers of genes that they represent may not correspondingly increase. Therefore, the two orders of magnitude difference in bacterial genome sizes from the smallest bacterial genome to the largest bacterial genome is a fair representation of the scale of difference in the information that these DNA molecules contain. So this means that the smallest bacterial genome, which is about 100,000 bases, may code for about 100 genes, whereas the largest bacterial genome, for 12 or 13 megabases in size, would code for about 12,000 odd genes. So the difference in genome sizes translates to difference in the number of functional genes. So apart from gene numbers, if I were to look at the universe of all gene functions that I know of and ask how common are these gene functions across the bacterial genomes that I have sequenced, I'll find that there are very, very few gene functions are common across all or most bacteria, and that most gene functions are unique to small subsets of bacteria, which is again yet another measure of genetic diversity. Okay, and then I'll argue, oh well, I'm talking about all of bacteria which have existed for whatever 3.5 billion years or something on Earth, and have undergone a lot of evolution, and we've already seen that bacteria occupy the pride of place in the tree of life. So why should I not be surprised? But then it turns out that even within a bacterial species, for example, a famous bacterial species such as E. coli, we find that even within this species, there is extraordinary genetic diversity. Within this species, the smallest genome and the largest genome can show as much as 25% difference in just their sizes. And again, if I were to look at gene functions that are found in E. coli, across E. coli, and ask what proportion of these are universally found across all E. coli, I'll again find that it's only a very, very small proportion of gene functions that are conserved across all E. coli. And any new E. coli genome that I sequence today is likely to discover some tens of genes that have not been seen in any other E. coli genome sequence thus far. And this diversity in gene sequence in E. coli translates to the extraordinary flexibility of this species in being able to inhabit diverse environments. And even among E. coli that inhabit humans, you will see that some are harmless and probably useful components of the human microbiome. Some cause disease of the intestine, and some cause diseases of the urinary tract, and even within these diseases with different manifestations. So even within a species, we can see extraordinary genetic diversity. Now, what is the source of this genetic diversity? And pretty much anybody doing biology will talk about the process of mutation. Mutations happen in uh, um, the dogma is that the mutations are random, and then selection will act on these random mutations and then sort them out so that the fitter mutations take over the populations. It's not strictly true. Mutations need not be random. There is quite a bit of non-randomness that is built into the way mutations occur, and that any molecular biologist would be able to will be able to tell you that. We will not get into that so much, but instead we'll also talk about the processes of gene loss and gene gain. Now, as I said, the genome's size spans two orders of magnitude, which means that I start off with some organism with a certain genome size, and as evolution proceeds, these genomes should either grow in size or shrink in size, and that means they should be either gaining new DNA or losing the DNA that they already have. And various studies show that um, um, losing DNA is sort of a continuous process that happens in bacteria and probably also in eukaryotes, but that genomes just don't disappear to nothingness because of some combination of selection, as well as um, sporadic bursts of gene gains that happen on and off. Now, gene losses dominate the evolution of certain types of organisms, which end up being endosymbionts. These are endosymbionts are bacteria that live inside other organisms or even inside other larger cells, and they are sort of in a very, very cozy environment and do not have much competition from any other organisms. And sitting in this environment, they are happy to lose anything that is not essential for their very survival. And these turn out to be very, very small genomes, just a few hundred kilobases in length. And on the other hand, when we look at genome growth, then we start seeing some interesting things. Um, Again, the textbooks will tell us that new genes are gained by a process of gene duplication and so-called neo-functionalization. That is, an existing gene duplicates, and when you have two copies of a gene, the selection on maintaining the sequence of one of the two copies decreases, and that copy now is free to evolve into something else, and then you produce a different gene. This kind of duplication processes seem to be important in the growth of eukaryotic genomes, but they seem to play a much smaller role in the growth of bacterial genomes. It turns out that bacterial genomes instead grow through this phenomenon called horizontal or lateral gene transfer, by which they acquire DNA not from their parents, which is the normal mode of um, 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 reproduction, but instead they gain DNA from other organisms that may be living along with them, even from a bacteria that may be a very, very different species from even archaea or through even viruses. So, this process of horizontal gene transfer appears to be important for genome growth and seems to underlie the expansion of large numbers of gene families. Now, this is responsible for bacteria acquiring different kinds of properties, and these include virulence. A classical example for this is the gain of uh, a protein called the cholera toxin in the bacterium Vibrio cholerae, which gives Vibrio cholerae the ability to cause cholera. And in fact, the evolution of Vibrio cholerae from a non-pathogenic version into different um, pandemics of cholera that have happened over the last 150 years or so is driven by horizontal gene transfer of different virulence elements. And this kind of horizontal transfer is probably mediated by bacteriophages or viruses that prey on bacteria. Yet another a property that is driven by horizontal gene transfer is the gain of antibiotic resistance, especially in the so-called escape pathogens such as E. coli, Klebsiella, Acinetobacters, and so on. And these are essentially genes that are able to break down or throw out antibiotics from the bacterial cell. And these are present in some bacterium in the environment, and other bacteria can acquire these DNA horizontally. And Horizontal gene transfer is responsible not only for these kinds of accessory properties such as virulence or antibiotic resistance, but even fundamental metabolic processes, including gene components that play a role in something like bacterial photosynthesis, can expand, diversify, and be transferred through transfer, horizontal gene transfer. Now, finally, if a horizontal gene transfer and horizontal gene transfer from some one arm of the phylogeny into some very distant branch is so common, uh, does the idea of a phylogeny itself make sense? It turns out that in spite of all these horizontal transfers, the genetic information of these organisms still retain sufficient signatures of parent to child vertical genetic transfer that a phylogeny built out of um, an assumption of vertical transfer still holds true. So, what you instead have is a phylogenetic tree, which is essentially a branching a tree determined by vertical transfer, on which you add an extra layer of transfers from one branch to the other, like probably trying a thread from one branch of a tree into another. So you have a tree over which a network is overlaid, and so, in a summary, the idea that I've wanted to convey is that bacteria are a very, very dominant form of life on Earth, without which probably other forms of life wouldn't really exist. And these represent extraordinary trait diversity, which correlates very nicely with, again, extraordinary genetic diversity. And this genetic diversity is caused by pretty much any mechanism that you can conceive of. It's normal processes of mutations, losing DNA, gaining DNA not only if, by duplication processes, but also voraciously taking up DNA from the environment or from other bacteria. So this is essentially the bacterial world that I wanted to talk about. Thank you.